I'm going to discuss about the amino acids and the proteins. And this will include the basic to the higher level of protein and its structure understanding. Now, the key aspects of central dogma, as you have seen in the earlier classes, that you could see the information flow, that is genetic information primarily flows in one direction from nucleic acid to protein formation, and then replication, and then you can understand this in terms of in the figure way, that is from DNA to RNA, and from RNA the protein is translated. So transcription and then translation, the codons are formed from these RNAs, and then these codons are responsible for the amino acid formation. The details of the amino acid connection one by one with each other has also been discussed in the earlier class. So here you could clearly see the different amino acids are formed here, and as a result, these is- amino acids are further connected one with another, and this leads to a formation of long peptide. Now, the same thing can be clearly understood here, that this is DNA, and then DNA forms the RNA, that is transcription, and then these RNA three sets of bases forms the amino acid, and these three sets are of different combinations, and different sets of these bases or codons are responsible for the formation of different amino acids. And this is what you can see as a symbol like V, H, L, T, P, E, E, and K. So now it is coming that the six sets of what are the sets of bases which forms the amino acids. So here there are 64 sets. Out of 64, one is responsible for methionine, which is kind of initiation codon, and then three are responsible for stop signal, that is a stop codon, and the rests are responsible for all the amino acids, and this is clearly presented here. Now, so as you understand that DNA is located in nucleus, then RNA is located depending on the type of RNA in maybe nucleus as well as the cytoplasm. And finally, the protein is definitely formed in the cytoplasm, as you can see in the form of polypeptide. Now, so what is protein? So you need to understand the protein structure. So basically it provides the structure, catalyze cellular reactions, and carry out many other tasks. And it has multiple biological functions classified. These proteins are classified according to their biological roles. Now, types of proteins and their functions. For example, enzymes, mostly varied and specialized proteins with catalytic activity. All chemical reactions of organic molecules, biomolecules in cells, are catalyzed by the enzymes. Now, second protein, transport proteins. So it is helpful in transportation. For example, they bind and carry molecules or ions to the organs in blood plasma. Lipoprotein in the blood plasma carries lipids from the life to other organs. Now, nutrients and storage proteins. For example, seeds of many plants store nutrient proteins required for the growth of the germinating seedlings. Oval bumin, the major protein of egg white, and casein, the major protein of milk, are examples of nutrient proteins. Now, contractile or motile proteins. Some proteins, endo cells and organisms with the ability to contract, to change the shape, or to move about. Tubulin is the protein from which microtubules are formed or built. Now, structural proteins, many proteins serve as the supporting filament, cables, and or sheets to give biological structure, strength, or protection. Major component of tendons or and cartilage is the fibrous protein collagen, which has very high tensile strength. Ligaments contain elastin, a structural protein. Now, defense proteins defend organisms against invasion by other species or protect them. Immunoglobulin or antibodies are made by the lymphocytes of vertebrates and can recognize and precipitate or neutralize invading bacteria. Fibrinogen and thrombin are blood clotting proteins. Now, regulatory proteins, it helps in regulating cellular or physiological activity. The cellular response to many hormonal signals is often mediated by a class of ATP binding proteins called G proteins. Then it comes to amino acids. So, which building blocks of proteins. Every amino acid has the same fundamental structure, which consists of a central carbon bonded to an amino acid group, a carboxyl group, and a hydrogen atom. Now, example of an amino acid. Here you can see R group, that is side chain here, central carbon, which you can see here, then amino group and a carboxyl group. So next, amino acids, side groups. There are 20 different types of amino acids used in making proteins. You can change or you will have a change to study them interactively on the websites, on different websites. The essential structure is the same in all the 20 amino acids or molecules. The 20 molecules differ in the side groups only. Now, amino acids, here a polypeptide bond is a covalent bond formed. Polypeptides are polymers of amino acids linked together by peptide bonds with a amino group of one acid joining the carboxyl groups of its neighbor. Now, here you can see how they are linked with each other. So, amino acid, two amino acids are there. Now, dipeptide, here you can see how they are formed between these two amino acids. Now, amino the peptide bond is formed when the OH group from the carboxyl group of one of the amino acids is removed and a hydrogen from the other amino acid is removed, the OH and H bond to form the water. The formation of water from the two amino acids is called dehydration synthesis. The covalent linkage that is formed is known as a peptide bond, and the molecule that is formed by the linking of amino acids is called polypeptide. Now, proteins are very large molecules. Some proteins consist of a single polypeptide chain, and others called multi-subunit proteins have two or more domains. Then individual polypeptide chains in a multi-subunit protein may be identical or different if at least some are identical. The protein is sometimes called an oligomers protein, and the subunits themselves are referred to as promoters. Now, labels of organizations of the proteins. Now, primary structure, secondary structure, and quaternary structure. So primary, secondary, tertiary, and quaternary structure, you can see the primary structure here, completely a linear formation. Then secondary structure, you can see in the form of sheet. Right here, alpha helix and B pleated sheet. Then tertiary structure, you can see more complexity in terms of structure. And finally, the quaternary structure is also possible, which looks more and more complex than the tertiary structure. Now, the same level of organization can be seen. The primary structure, here you can see completely linear. Then secondary structure in the form of sheets, which is clearly available here. Then you can see the tertiary structure, and finally the quaternary structure. The same thing could be described a bit. For example, primary structure, amino acids bond with each other. So here with the help of peptide bonds. And then secondary structure, alpha helix and beta sheets, you can see. Tertiary structure, you can see how they are formed, and quaternary structure, highly complex structure, much more advanced than or complex than the tertiary structure. So the protein structure can be divided into four main categories: primary, secondary, tertiary, and quaternary. In primary structure, sequence of a chain of amino acids are present. Now, secondary structure, hydrogen bonding of the peptide backbone causes the amino acids to fold into a repeating protein pattern. So, the tertiary structure, three-dimensional folding pattern of a protein due to side chain interactions. Quaternary structure, protein consisting of more than one amino acid chain. Now, how you can define the primary structure? Covalent bonds between amino acids and is normally defined by the sequence of peptide bonded amino acids and locations of disulfide bonds. Now, the relative spatial arrangement of the linked amino acid is unspecified. Primary structure is the linear sequence of amino acids in a polypeptide chain where amino acids are linked with one another with the help of peptide bonds. This specific order of amino acids is determined by the organism's genetic code and is crucial because it dictates how the protein will fold, ultimately defining the three-dimensional structure and the function. A change or mutation in even one amino acid can significantly alter the protein's overall shape and the biological function. Now, you can see the key characteristics of amino acid sequence. The core of primary amino acid primary structure is the unique order of 20 different types of amino acids that form the polypeptide chain. Then peptide bonds, these strong covalent bonds hold the amino acids together in the chain and terminus to C terminus. The sequence is covalently written from amino terminal, that is N end, to the carboxyl terminal or C terminus end. Genetic basis, the primary structure is encoded in an organism's DNA, which is transcribed into messenger RNA or mRNA and then translated by ribosomes into the amino acid sequence. Now, what is the importance of primary structure? It determines function. The specific sequence of amino acid is the fundamental blueprint that dictates how the protein will fold into complex 3D structure and which directly determines the protein function. Impact of mutations. Genetic mutations can change the DNA sequence, leading to the incorporation of a different amino acid. This single change can have significant effects on the protein structure and consequently its function, as seen in conditions like sickle cell anemia. And here you can see loss of critical core hydrophobicity, mutation critical surface site, increased clashing, and decreased matching residue pair. That here you can see misfolding. Now, folding, you can see maintained critical core hydrophobicity, intact critical surface areas or surface site, increased matching and decreased clashing residue pair, here folding or perfect folding. Now, secondary structure, it refers to regular recurring arrangements in the space of adjacent amino acid residues in a polypeptide chain. Then there are few common types of secondary structure, the most prominent being the alpha helix and the beta sheet conformation, which you can see here how the alpha helix could look like and beta sheet or beta pleated sheet would look like. Both the patterns are shown here. Then protein secondary structure refers to the local folding patterns within a polypeptide chain, primarily the alpha helix or a spiral and beta pleated sheet, a sheet-like structure, which are stabilized by hydrogen bonds between the polypeptide backbone atom. These regular repeating arrangements provide the protein with mechanical stability and form the building blocks for more complex three-dimensional protein structure. Main types are alpha helix and beta pleated sheet. Alpha helix, a right-handed coiled structure where the polypeptide chain twists into a spiral. Hydrogen bonds form between the hydrogen atom and one of the amino acids amide group and the oxygen atom of the carbonyl group of an amino acid 4 residues away in the helix. Beta pleated sheet is structure made of multiple relatively straight strands of the polypeptide chain that lie side by side, forming a sheet-like arrangement. Hydrogen bonds form between these adjacent strands. Now, how they are formed. Hydrogen bonds, both the alpha helix and the beta pleated sheets are stabilized by hydrogen bonds formed between the backbone atoms of the polypeptide chain, specifically the carbonyl oxygen and the amide hydrogen of different amino acid residues. Backbone interactions, these bonds involve the repeating sequence of amino atoms in the polypeptide backbone and not the side chains of the amino acids. Other secondary structures are also possible. While the alpha helix and beta pleated sheet are the most common secondary structures, but there are also other secondary structures which could exist and which exist. Beta turns, structures that allow the sharp turns in the middle of a polypeptide chain, that is beta turns. Omega loops, another type of loop structure. Triple helix, a common secondary structure found in collagen, a connective tissue protein. Random coil, segments of the polypeptide backbone that are not part of regular repeating structure. Now, the secondary structure in terms of the detailed structure of each amino acid and the binding possibilities, you can see clearly with the detailed amino acid and bound with each other in case of secondary structure. Now, secondary structure, tertiary after secondary structure, now this is the most important type the of the amino acid, sorry, proteins, that is tertiary structure, the spatial relationship among all the amino acids in a polypeptide chain. It is the complete 3D structure of the polypeptide. And this 3D structure diagram illustrates some of the key bonds present in the tertiary structure, such as hydrogen bond, hydrophobic interactions, polypeptide backbone, then disulfide bridge, and ionic bond. These three bonds, hydrogen, disulfide bridge, and ionic bond, you need to understand very well. Then how the hydrophobic interactions are possible, and then polypeptide backbone. Next, here you can see the types of protein, but the tertiary structure is located here. So this is the primary, secondary, and tertiary, and finally the next type of structure, that is quaternary structure. Now, tertiary protein structure is the overall 3D structure, a 3D shape of a single polypeptide chain formed by further folding and coiling of secondary structures like alpha helices and the beta sheets. This unique 3D configuration is stabilized by various interactions between the amino acid side chains, including disulfide bonds, ionic bonds, hydrogen bonds, and hydrophobic interactions. The tertiary structure dictates the protein's final functional form, and misfolding can lead to different diseases. Key aspects in 3D structure of the protein which you need to understand is the 3D shape. It describes the complete complex shape of a single polypeptide chain in a space, packaging local secondary structures into globular region called protein domains. Stabilizing interactions. The folding process is driven by interactions between the R groups and the of the amino acids. Now, hydrophobic interactions, non-polar side chains clustered together in the interior of the protein away from the surrounding water, forming a hydrophobic core. Hydrogen bonds occur between the polar side chains and the backbone atoms, or between polar side chains themselves. Then, ionic bonds or salt bridges form between oppositely charged acidic and basic amino acid side chains. Then it comes the disulfide bonds, covalent bonds between the sulfur atoms of the cysteine residues, which are strong and rigid. Now, the functional unit for proteins consisting of a single polypeptide chain, the tertiary structure is the final functional form. Misfolding, incorrect tertiary structure can rise from mutations in the primary structure and can lead to a loss of function and finally to the diseases. Types of proteins. Proteins are often categorized by the tertiary structure into either compact spherical globular proteins or elongated fibrous proteins. Then here, how you can see the impact of the change in different level of protein structures and the diseases. For example, normal and single cell. You can see the primary structure here and then mutation in the site of valine. Then here, this is valine. So sixth amino acid here is something else. Then secondary structure and tertiary structures. Normal subunit, but sickle cell beta subunit is somehow different in case of sickle cell anemia, which you can see the sickle cell hemoglobin. Then, in terms of function, proteins do not associate with one another each carries oxygen in case of normal condition. Then, in case of sickle cell anemia, here you can see that proteins aggregate into the a fiber capacity to carry oxygen is reduced. Now, it comes to the quaternary structure. Proteins with several polypeptide chains have one more level of structure referred to as the quaternary structure. It refers to the spatial relationship of polypeptides or subunits within the protein. Quaternary protein structure describes the final 3D arrangement of multiple folded polypeptide chains or subunits within a single functional protein complex. These subunits, which can be identical or homomultimers or different heteromultimers, are held together by non-covalent interactions like hydrogen bonds, hydrophobic forces, and ionic bonds, and sometimes by disulfide bonds. A classic example is hemoglobin, composed of four subunits that work together to transport oxygen. What are the key features? In case of quaternary structure, multi-subunit assembly quaternary structure only applies to proteins composed of two or more polypeptide chains. Non-covalent interactions. Subunits are linked by weak forces such as hydrogen bonds, hydrophobic interactions, and ionic bonds. Then covalent bonds, sometimes disulfide bonds, though less common than a tertiary structure, can also stabilize the quaternary interactions. Functional complex, the overall arrangement of these subunits creates a complex with a specific biological function. And here you can see how the primary structure to secondary and tertiary, and finally the quaternary structure goes on. Now, how it differs from the other structures. So it is clear that there are fundamental differences from primary to quaternary structures. The linear sequence of amino acids, that is primary structure. Local folding patterns such as alpha helices and beta sheets formed by hydrogen bonds along the polypeptide backbone, that is secondary structure. In case of tertiary structure, the overall 3D shape of a single polypeptide chain resulting from the interactions between amino acid side chains. Quaternary structure, the final arrangement of multiple folded polypeptide chains or subunits into a functional protein complex. And the same thing could be encaptured in this figure that is clearly seen. Now, the examples of quaternary structures, that is a well-known example is hemoglobin, that has a quaternary structure consisting of two alpha and two beta globin subunits. Each subunit contains a heme group where oxygen binds. Oligomer enzymes. Many enzymes in the cell are oligomeric, meaning they are composed of multiple subunits that come together to form an active complex. Finally, you can also summarize the entire comparative analysis between primary, secondary, and tertiary structure. Here, primary structure, linear sequence of amino acids. Secondary structure, folding of the peptide chain into a alpha helix or beta sheet. Then 3D structure of a protein, that is tertiary structure. Primary structure is linear, while secondary structure is either an alpha helix or beta sheet, and tertiary structure is globular. Primary structure composed of peptide bond formed between amino acids, while secondary structure encompasses hydrogen bonds. Tertiary structure encompasses disulfide bridges, salt bridges, and hydrogen bonds. Formed the primary structure formed during translation, while secondary structure forms collagen, elastin, actin, myosin, and keratin-like fibers. And tertiary structure includes enzymes, hormones, albumin, globulin, and hemoglobin. Primary structure involved in post-translational modification. Secondary structures are involved in forming structures such as cartilage, ligaments, and skin, etc. Tertiary structure are Tertiary structures are involved in the metabolic functions of the body and signal transduction process. That's all for the final and summarized short presentation for protein structure and the building blocks